Hello, everyone.
Thank you.
My name is Kathryn Elmer. I am a master's student at McGill University studying with Dr. Margaret Klaska. And today I'll be talking to you about remote sensing of invasive Phragmites. And you might have heard some of this before from previous meetings, but this is kind of an update on the new work that I've been doing. So just as a reminder for people who are new or maybe don't quite remember, the species of interest for my project is Phragmites australis, subspecies australis, which is the invasive version of the Phragmites reed. Here in the picture, you can see it grows quite tall. This is actually a 2-meter survey pole, and it grows quite densely, so it tends to choke out other vegetation that might be growing in that ecosystem with it. So it's a major species of concern. So since I'm the first one of the day, I'm just going to do a brief 30-second crash course in remote sensing, since there will be some other talks that discuss remote sensing as well. So the basic idea is that you have a sensor, in this case a hyperspectral sensor, which flies over an area and collects essentially a data cube. So you have a spatial domain and a spectral domain, where basically each pixel contains spectral information for whatever target falls within that pixel. So here, for example, we have a pixel of grass. You can see its reflectance profile. So my study area that I had been working in was Îles-de-Boucherville National Park, which is an island in the St. Lawrence River just outside of Montreal. And so last summer, I spent 2 weeks collecting field data within the park, which consisted of 320 GPS points, where they were collected as ground control points for future target detection analysis, which I'll discuss later. So each GPS point was collected and contained photos of the point that was measured. And I measured them in order to capture a variety of the different targets and materials that are present within the park. For example, you can see in the bottom middle picture, there was some dirt, as well as different types of vegetation, such as goldenrod, phalaris, grass, Phragmites, etc. And so the goal of this was to kind of cover the entire extent of the park with these points, which would then be used to validate target detection attempts while trying to identify Phragmites. There were some areas in the park which are marked by the purple and red hash mark boxes that I was not actually allowed to go in to collect points. But otherwise, you can see the spread. It was a relatively even split of those 320 points between Phragmites and non-Phragmites. So the other aspect of my project is the hyperspectral imagery, which is flown using the Twin Otter aircraft from the National Research Council of Canada, and on board it carries a CASI 1500 hyperspectral sensor, which collects data over 288 bands ranging from 366 nanometers to 1053. So last summer in 2019, the Twin Otter went over Îles-de-Boucherville and captured 5 flight lines that covered the entire extent of the park. Here you can see the green lines are showing the flight path of the aircraft over the park. And on the right side, you can see, 21, 22, those are flight line designations. And so later I might refer to specific flight lines by those number designations. So just something that I want to touch on briefly is one of the steps that we take when pre-processing hyperspectral data for end users to use in their analysis is that CASI pixels aren't actually square, they're rectangular. And this is something that Deep will be talking about in his presentation later in the day. So if you have any questions, he would definitely be the right person to ask. So we have this rectangular pixel, and previously what we had been doing is we would sum these pixels in the across track direction. And so you would have your line of pixels here, and you would sum each two together, but that still gives you a rectangular pixel. And so what that can cause is that it has an increased rate of pixel duplication. You can have higher false discovery rates because you're having pixel duplication. So targets that are maybe only in 1 pixel would now appear to be in 2. So one of the new things that we had been doing, because our data is processed specifically for the chosen application, which in my case is target detection of Phragmites, is rather than summing by 2, summing by 5. So again, you have your row of pixels here, and it's, I'm not sure why it's doing that. There we go. So instead of summing by 2, you sum by 5. And so what that actually gives you is a pixel that's actually more square. And so this can have a couple benefits to it, as well as the fact that you have a decrease in the amount of pixel duplication. Therefore, you have a lower false discovery rate. It also can have less sensor-induced blurring, a higher signal-to-noise ratio because you're actually including 5 pixels worth of signal in one. However, one of the trade-offs is that you do have a lower spatial resolution. So rather than a pixel being 1 meter by 2 meters, you're now 2 meters by 2 meters. So this is actually the flight line from 2018. This was the first data set that I had kind of been working with, and this is summed by 2 in the across track direction. And what you'll notice is that there were only 3 flight lines, so actually portions of the park were not captured in the data collection, which actually meant that some of the ground control points I had collected ended up being basically cut out of the usable area of data. However, in 2019, we got a much clearer data set. The 2 extra lines, so 5 lines total, really helped capture the edges of the park, as well as it's summed by 5. So this data set is a bit more appropriate for my target detection question because of the method I had just described about summing by 5. So this is the data set I had been using. And so running some target detection analysis, for example, this is the upper portion of line 24 in the park using the spectral angle mapper method. So the red indicates Phragmites. And so you can already kind of see it growing here in the agricultural ditches between fields, as Phragmites loves to grow in disturbed soil. Here's a little bit more of a zoom in to the line. And so the red again is Phragmites. And here are some of those ground control points that indicate the presence of Phragmites that I had measured. So you can already see that while it looks good, there's still some work to be done. A lot of times the target detection method gets a little confused in areas of forest and also in areas of the road. So that's something that does need improvement. Here's the lower portion of that same line. And so you can see as well here that there's maybe some confusion in the parking lot as well as some confusion in the trees. So from this point, it's trying to refine this target detection methodology in order to increase the accuracy of the results. But so far, it's showing that it is capable of picking out Phragmites. So it's refinement moving forward. One of the things that I had noticed working with the imagery is that the lower portion of Îles-de-Boucherville seems to be more complex than the upper portion of the park. The upper portion has a lot more of very clearly delineated agricultural fields, whereas the lower portion has a lot more kind of like mixed fields, mixed vegetation and such. So one of the things that might need to happen is a different target detection approach when handling the upper portion of the park versus the lower portion. So what I had been doing was I would take an entire flight line, as you can see on the left there, and I would actually just split it in half to deal with the upper and the lower portions. But you have to do that 5 times. So you end up with quite a few segments of data. So one of the things that I had been discussing with Margaret and with others is actually finding a more ecological way to explain that stratification between the upper park and the lower park, and kind of taking a different approach to addressing that in the target detection method. So one of those would be, rather than to separate individual lines, is to actually separate the whole upper portion from the whole lower portion once that stratification can be justified correctly. And then mask out areas of known no Phragmites. So like we know it's not in the, you can't really see it in the forest. It's not on the road. And so masking those areas out, because then what that does is it reduces the noise in the data set for target detection, which can help improve the accuracy of the target detection methods to identify Phragmites. And then additionally, these larger subsets of the data would increase the number of available ground control points for accuracy verification. Whereas before in the individual lines, you obviously can't use the entire subset of ground control points in that one little piece of the line. So some of the other next steps is, other than addressing that stratification question with the data, once I have the classification results, going into ArcMap. And one of the things that I was really interested in is what is kind of the smallest stand size of Phragmites that this type of approach could correctly identify. And so the way I'm going to answer that question is to basically take that classification image, turn it into polygons within ArcMap, and then create a graph of polygon area versus frequency. So is it more common for there to be larger areas of the Phragmites detected or smaller areas or a mix? And that might help to answer that question of, you know, could you you know, when your pixel is 2 meters by 2 meters, are you able to detect Phragmites if it's a sub-pixel stand size, or does it need to be larger than that in order to be correctly identified? So kind of following on that, generating statistics for that classified Phragmites, you know, calculating total area and so on, and then generating that final classification map of Phragmites for the entire park. Possible future improvements that I think would be really interesting to apply would be the inclusion of LiDAR data sets. Phragmites usually grows to be much taller than the vegetation that it's around. And so I think it would be really interesting with LiDAR because you could see the ground height, you could see the Phragmites stand height, and maybe that is something that could help refine this target detection method by taking into account the height of the stand. Similarly, digital surface models might be able to kind of reveal that same information about the height of Phragmites stands. And it also might highlight, you know, growing preferences for Phragmites. You know, in the literature, it says that they like low spots, they like a certain level of soil moisture. And so maybe by adding these things into that target detection methodology, it might further be able to refine the results and the accuracy of identifying Phragmites using remote sensing. So thank you very much for your attention. I'm happy to answer any questions that you might have.
Thank you very much, Kathryn. It was a really good presentation. So if you have questions, you can open your microphone or yes. So we have a question from Crystal Schaff. You can open your microphone, Crystal.
Thanks. Very nice, Kathryn.
Thank you.
So yeah, we've done some work over Plum Island with airborne LiDAR, fairly coarse G-light, which is more like 3-4 meters, but certainly the Phragmites pops right out. I can send you that paper.
Oh, sure. That would be really interesting to read. I think that that would be kind of a cool data fusion to apply that LiDAR to this question. But yeah, I would appreciate that. Thank you.
I had a question.
Mark has a question too.
Oh, sorry. I didn't raise my hand.
Oh, sorry.
No, go ahead. I didn't raise my hand. Frank first.
I think co-host can't raise their hand, so you can just type in the chat that you want to speak. So Mark has a question and then Etienne.
Well, I will be quick because other people had their hands up before me, but I have the microphone. So anyway, so I guess I was trying, as a non-remote sensing person, I was trying to imagine what would happen, like what would have happened if you'd done all the same analysis with like a 43 centimeter by 2 meter pixel. Like I kind of get the pros and cons, but what, like, if you had done that, what do you think you would have found? Like too many false?
Yeah, I mean, without actually running it, I think it's hard to say exactly, but I would expect that there would maybe be a higher false discovery rate of Phragmites due to the fact that once it actually goes through all those pre-processing steps, in order to fill in some of the blank spots in the data, it does have to duplicate pixels. And so having those rectangular pixels means that there is more duplication. So I would expect either for there to be, you know, more Phragmites than there, or more classified Phragmites than there really is, because it might be taking a Phragmites pixel and duplicating it, and now you have twice as much. Or also, you know, the contribution of pixels around it that maybe don't have Phragmites, well, now those are maybe getting duplicated and, you know, kind of covering up a pixel that might have Phragmites in it. And so now maybe you're not actually seeing all of the Phragmites that would be there. And so that's why kind of taking the question that's being asked into consideration while doing those pre-processing steps kind of helps ensure that you get a data product that is most appropriate, I guess would be the best word, appropriate for the question that you're trying to answer with it.
Yeah, that makes really good sense. The second one I think would be of concern. The first one, like if a pixel says, if you get a signal that says there's Phragmites here and there really isn't, but it's right next to it, that doesn't really concern me so much. I mean, we're not, right. But actually to get a signal that it was there when it wasn't, that's a little more.
Right, and again,
it depends on proximity too.
Right, and that actually comes down to the question that's being asked. And so that was something that I had to consider when first kind of planning this study, was like based on the literature and based on talking to the park management people at Îles-de-Boucherville, it kind of comes across as like you don't want to be going all over the park chasing down kind of a phantom Phragmites signal. But at the same time, because it is so prolific, because it is so invasive, you know, there's a certain amount of, you know, like you said, if it's an extra pixel on the edge of something that really is Phragmites, you know, they're going to be there probably looking at it anyways, but it's trying to draw that line to make sure that you're not sending those field teams running all over the park and then they get there after 30 minutes of trekking off the trail and it's like, there's nothing here, you know. So that is something that I like have to be aware of when doing this type of analysis for sure.
Thanks.
Thank you.